Hi students, today we are going to discuss about multiple alleles. Okay, so multiple alleles. What is multiple alleles? What is the role of the multiple alleles? All these things we will be clearing at the end of the sessions. If you like my channel, press like button and share with your friends and subscribe it too. What is multiple alleles? Usually, a trait can be controlled by a pair of alleles. Am I right? Allele of a gene. Okay, one gene, one trait. One gene, one trait. So this trait controlled by a pair of alleles. A pair of alleles. A pair of alleles control a trait. In case of multiple alleles, 3 or more alleles of a gene can control a particular trait. Got it? 3 or more alleles of a gene can control particular trait. These alleles known as multiple alleles. And the inheritance of multiple alleles is known as multiple allelism. Okay, what is the example for multiple allelism? The basic example is ABO blood groups in human beings. ABO blood groups. Okay, ABO blood groups, the best examples. So how we can say ABO blood groups the best example for multiple allelism? Okay, so we have different. Each persons have their own type of blood group. Some will be having the blood group A, some person will have blood group B, some persons will have blood group O. Okay, based on their chemical differences found on the surface of the erythrocytes. The erythrocyte, that RBC, on its surface contains a particular type of antigens. Based on that antigens, we say the person is having A blood group or B blood group or O blood groups. Okay, if this RBC does not have this antigen on its surface, then it is termed as an O blood groups. Do you understand what I am saying? So everything because of this RBC on its surfaces, it is having the antigen. So this is, we call it an antigen. If it is having antigen A, then this blood group, A blood group. If that RBC having antigen B, then it is known as B blood groups. If it is not having, means then it is an O blood group. Do you understand what I am saying? If the RBC on its surface has antigen A, then this blood group is termed as an A blood group. If it is having antigen B on its surface, then it is considered as a B blood group. If it is not having any antigen on its surface, is considered as an O blood groups. So this blood group is based on the chemical differences found on the surface of the erythrocytes. Am I right? Clear? Let's go a little deeper enough to understand. If RBC is having both antigen A and antigen B, antigen B, then it is considered as an AB blood groups. Okay. If A blood group always get blood from the A. B blood group always get blood from the B. A can donate to only A. B can donate to only B. If A blood groups receive B blood groups, A blood types receive B blood types means what will happen? And the erythrocyte will occlusinate. Erythrocyte will be occlusinating. Okay, so leading to the death. So that's why. So when we are donating the blood, and they will be first checking out what type of blood group it is. So A blood groups will get blood from the A blood group persons. Okay, the same B to B. Okay, if it is given incompatibility of blood groups, then leading to the occlusions of the erythrocytes, that leads to the death. Okay, fine. O is considered as an universal donor since it is not having antigen A, antigen B on its surfaces of erythrocytes. So it is considered as a universal donor. Okay, AB blood group is considered as an universal acceptor since it is having both antigen A and antigen B, and it is able to accept blood group A as well as blood group B and O. So that's why it's an universal acceptor. AB blood group is an universal acceptor. Okay, fine. So let's move on. Who has identified blood grouping? Okay, who has proposed this blood grouping? Carl Landsteiner. Okay, is a biochemist. Carl Landsteiner is the biochemist. He has identified A blood groups and B blood group. These two types of blood groups are identified and informed by Carl Landsteiner, who is the biochemist. Followed by him, his students identify Van de Cassis and Sterling. These two students of Carl Landsteiner identified and discovered AB blood group, which is a rare blood group. Rare. The gene which is responsible for blood grouping, or the gene which controls the blood grouping, is termed as L. Is termed as L. The gene is termed as L. The gene which controls the blood grouping is termed as L or I. What is the meaning of L? Stand for Landsteiner, I stands for isoagglutinations. I stands for isoagglutinations. This gene L or I has three allelic forms. Okay, they have three types of alleles. One allele A, allele B, allele O. These three types of allelic forms are responsible for the blood group control. Okay, so what is the allele A will do? Allele A encode. What it is encode? Encode enzyme for okay, so allele A encodes N-acetyl galactose transferase. Okay, it can add up. It can add NAG. It can add NAG. What is NAG? N-acetyl galactosamine. N-acetyl galactosamine. So IA gene encode N-acetyl galactose transferase. N-acetyl galactose transferase. What does allele B encode for it? Allele B encodes enzymes for galactose transferase. Galactose transferase. Okay, so allele B encode for enzymes galactose transferase. It can add up. It can add. What it can add? Precursor. Galactose precursor. It can add up galactose to the precursor. Understand? Understand? IA encode. IA gene encode for N-acetyl galactose transferase. It add up with NAG. N-acetyl galactosamine. So IB code for the galactose transferase and it can add galactose to the precursors. So IA and IB encode for transferase function. Encode for transferase function. Got it? IO, since it is not having antigens and does not encode any enzymes, so thereby IO is null. Okay? IO is null. Does not encode transferase enzymes. Do you understand? The main function of the IA, IB and IO alleles. IA alleles encode N-acetyl galactose transferase. It can add N-acetyl galactosamine. Okay? Then IB encodes enzyme for galactose transferase. It can add galactose to the precursor. And IA and IB together they do the function of transferase. Okay, IO does not have, this allele does not have any encoding process, so it is considered as null. Nothing is produced by IO allele. Got it? Okay, let's move on to the next level. Genotype. Okay. So genotype IA, it gives what type of blood group? Blood type will be A. Antigen is A. Okay. And this is also A blood. And this one is an B blood types. And this is also B blood types. And this is O, and this is A, B. So we are having the 4 types of blood groups. One is an A, B, O, and AB blood groups. These are our blood groups. Got it? So this IA alleles encodes blood group, blood type A, and the IA, since it is not having any encoding any enzymes, and this IA responsible for the blood type A, and it is also there's homozygous conditions, and this is also a dominance one is there, so thereby is able to give blood. IO gives B blood types, and IO gives O blood groups, and IB gives AB. Okay, and it can give possible blood group is A, possible blood group A, possible blood group B. Look at genotype IA alleles. So it is blood type is an A. IA allele blood type is A. The antigen found on the RBC is antigen A. The antigen found on the RBC is an antigen A. This allele IAIO, the genotype is IAIO, indicate the blood type A. The antigens found on the RBC is A. Okay, IB homozygous alleles, and it indicates the blood type B. The antigen found on the RBC is B. Okay, IBIO alleles indicate blood type B. The antigen found on the RBC is antigen B. Okay, in case of IO alleles, they don't have these antigens on the RBC surfaces. Okay, so neither they have O, or neither they have A, or neither they have B. Okay, so IO alleles, they indicate blood type O. Okay, here IA and IB alleles indicate blood types AB. Okay, so the antigen found on the RBC is antigen A and antigen B. So what will be the antibodies? Again, antigen A, or again blood group A. In blood group A, the antibody is anti-B. The antibody is anti-B. The same, the blood group A possesses anti-B. And blood group B will have the antibody anti-A. Blood group B will have the antibody A. Okay, and in case of AB blood group, neither they have anti-A or nor they have anti-B. So I hope so you understand the multiple allelism. Okay, so in case multiple allelism, 3 or more form of alleles of a gene control a particular trait. Okay, these alleles are present on the same locus of the homozygous chromosome of a gene. So these multiple alleles are present on the same locus of a homozygous chromosome of a gene. Okay, so thank you for watching my channel.